So thank you very much for this opportunity to talk here. Before I start, I would like to thank the warm welcome we had here at LPENS, in particular the genes, as we call them, Jean-Marc, Jean-Michel, Jean-François, for their warm welcome here, and the resource services, the Salle Blanche, the IT have been really helpful with us to answer our questions and help us. Right, so I'll talk about motility and cytoskeletal dynamics. Cytoskeleton, the cytoskeleton is a skeleton of a cell. And in fact, we have swimmers or crawlers. I gave you two examples here. This is a swimmer, you know, it's a spermatozoid, could be a bacterium. Here you have a crawler, so that crawls here on a substrate. This is a keratocyte. So the difference between swimmers and crawlers in living systems is that swimmers swim with a flagellum that is outside of the cell body, whereas crawlers crawl with what is inside of the cell body. So they push with biopolymers. But in fact, what I would like to say is that it's all the same thing physically. It's just filaments, biofilaments that assemble dynamically, either to propel or to push on from the inside of the cell bag. So in fact, we used to have flagella a long time ago. We were bacteria. We didn't have a nucleus. And then during development, we developed a nucleus inside our cells. And in fact, because we had to organize into tissues to be ourselves, so we had to get rid of this filament and use the filaments inside our body, our cell body. So in fact, before I start, I'd like to remind you what is the context of movement at the Say scale. So you know there is inertia. I think everybody has driven or seen a boat. When you stop the motor, the boat continues to drive. So the stopping distance is very large. So if you manipulate something very viscous, then the viscosity goes against the movement. So in fact, a way of knowing which one is dominant is to write the Stokes equation. And then if you consider an object of size A that moves at the velocity v in a system in a medium of viscosity eta and density rho, then you can estimate this term here, that is the inertial term. You can estimate this term here, that is the viscous term. And then you can define what is called the Reynolds number that compares initial forces to viscous forces. And so if this Reynolds number is very high, higher than 1, this means that inertia dominates, whereas if this Reynolds number is smaller than 1, this is viscosity that dominates. So if you look at Reynolds number for a swimmer, it's about 10 to the power 6. So I swim at a velocity of about 1 kilometer per hour. For fish, it's on the order of 10 to the power 3. And for bacteria or sperm cells, it's much smaller than 1. So the message, if you only remember one thing from this talk, it's that life at the Say scale is at low Reynolds numbers. So viscosity is the dominant factor, dominant force that moving systems encounter. So just because in biophysics, lots of people tend to do anthropomorphism. So I would like you to do this exercise of knowing in which conditions should you be put, in which hydrodynamic conditions should you be put to feel the same medium than our own sperm cells. So you just need to rebuild the Reynolds number, and then if you would feel the same environment as a bacterium or a sperm cells, you should swim in treacle, so very thick honey, and at 3 centimeters per hour. So it's just for you to feel what is the surrounding of a moving cell. So in the cell, the message is no inertia. So just to insist on that, so the time bacterium needs to stop is, the distance is 1/10 of angstrom, whereas the stopping time is less than a microsecond. So again, the only viscous force that is here encountered by the system is proportional to the velocity and the size of the object. Right, so now let's, now that we are all set in the conditions of these moving systems, let's look at the crawling, the cell crawling. So here you have a scheme that is now well verified, that when you put a cell on a substrate, so now one of our cells, this is the cytoskeleton inside the cell. So these are these biopolymers that organize. So the cell moves here towards the right. So how does it move? So underneath the membrane here, you have new biopolymer that have polymerized and that are able to deform the membrane, so to elongate this, what's called a lamellipodium. Then you have adhesion, so the cell adheres on the substrate, and the rear of the cell contracts by contraction of molecular motors on biopolymers. So in fact, I will not talk about adhesion at all, but I will talk a lot about actin polymerization and a little bit about this retraction. So if you look at these networks, these biopolymer networks here, they look like this in electron microscopy. So you see there are branched filaments here. And at the rear of the cell, you have the same sort of structure of branched filaments, but this time you have myosin motors that slide them, so that contract the rear of the cell. So we have worked on biomimetic systems of actin polymerization. And in fact, what inspired us was this intracellular movement. So inside the cell, you have some endosomes that are able to move by the same mechanism here of actin polymerization that pushes the membrane forward. Here at the surface, you have an activator of this polymerization in a branch network. And so these systems are able to move forward. And I will just briefly talk about the retraction. So in fact, what we decided to do, so it was more than 20 years ago, is because the cell is complex, the question was, can we mimic this to be able to control the parameters and understand the physics that is behind this movement? So our systems consist of a substrate, so it can be a bead, a droplet, a liposome, or anything, at the surface of which we graft this activator of actin polymerization. So we know that now that we make a branch network. So as you see here, so this protein here that is grafted at the surface is able to constantly branch this network that is growing. And so we can add, because the branching would go exponentially, we can add what we call capping proteins that will prevent the filaments from growing. So as soon as you have a new branch, you have a capping protein that can stop the polymerization of the filament to allow for it to branch more. And here I show you two movies. So one that we call symmetry breaking. So if I put this system around a bead, a spherical bead, this is spherical here. So because the new material new material growth occurs at the surface of the bead, then after some time it will break open. I'll come to that in a minute. And the next step is that beads are motile. So the motile is movement of a living organism. So beads move just by this actin polymerization at the surface. So in fact, the good news is that now the system is available, is commercially available. And Julie and myself recorded a Journal of Visualized Experiment paper. So you can just listen to the video, buy the proteins, and do it yourself in your kitchen. Everybody can do it, and we hope that it will be available for students at LPENS very soon. So you can just take beads, take anything, cover it with this activator of polymerization, and you can obtain these trajectories like here. So I will first describe what we understood in this symmetry breaking event. So in fact, it's, so I'll come to that later, but this network is elastic. It has elastic properties. So when you grow a first layer around this sphere here it's what I call the two-color experiment. So we start with red actin, and then we put green actin so that we can see which one is first. So you see that if we start with red actin, we have this network that grows. It's a shell that grows around the sphere. And if you add green actin, you see that it pushes away the already existing shell. So in fact, the monomers are added at the surface of the sphere here. So you have a first shell, and then the second shell pushes the first shell away. And so at the end, you have a tangential stress here that is built up during the growth of the network. So here, H is the thickness of the shell, R is the radius of the bead, and E is the elastic modulus. So in fact, if we put in the reaction at the surface, we end up with a steady state thickness that is proportional to the radius of the sphere. That depends on the chemical energy of polymerization, the Young modulus, here the mesh size of the actin network. We can always characterize this network as having a certain mesh size, and we can vary it. I will come to that later. And A is the size of a monomer. So in fact, when it breaks under this tangential stress, so we can estimate the release of the elastic energy, that's here, the cost of breaking bonds in the network. That depends on the fracture energy. Here is the energy per unit surface for one link. And then we have an energy balance that results in this sort of curve. So if we have a pre-existing crack size, if this crack size is larger than L star, we will generate a fracture. And then we have a critical stress at which it will fracture. And so which will correspond to a critical thickness at which the system will fracture. So all the question now is, how is the steady state thickness compared to this critical thickness of breakage? In fact, we have two regimes depending on the protein conditions. Either we have a growth of the actin network thickness that then breaks at some point that is before this steady state thickness is reached, or we have a growth that stays at this thickness, that is the elastic steady state thickness, and then at some point it will break open with time. So in fact, the distribution of time for breakage is very narrow in this case, is very large statistically in this case. So we have two cases. Either we have, this is just what I said, if we don't have a plateau, then this means that this critical thickness is lower than this one, and it's the opposite if we have a plateau. So we can vary. I won't go into the details, but in fact, here you see, because these two thicknesses depend linearly on R, we don't have a dependence of these cases with R. And you see that here, when we vary the radius of the bead, we are always in the case without a plateau, whereas here, we are always in the case with a plateau. So it's only the conditions, the protein conditions, that will allow us to vary these parameters here. So this is an example of this famous capping protein I was talking about before. So if we stop the filaments from growing once they have branched, then we have, you see that if we have a low, we have, so the effect of the capping of barbed ends increases this delta mu. So by increasing this delta mu, I can go from a plateau regime to a case 1 regime. Right, so now I will go to these trajectories. What can we derive from this movement? So, so first of all, so historically, so this is, well, in fact, there are bacteria that are able to move inside the cell by this mechanism, this actin comet mechanism that we see here. This is an electron micrograph inside a cell of a bacterium Listeria that is able to polymerize actin at its rear and so move within a cell. So in fact, this, the bead motility was reconstituted first in cell extracts. So cell extracts are cells, you break the membrane, so you end up with an inside of a cell that can be infinite, I mean, that can be high volume. And so you can put your beads in this medium and study them the same way, they behave the same way than inside a cell. So then bead motility was reconstituted in purified proteins. And I remember, so I will first show you this movie. This was our first movie, so I'm still emotional when I see this. It worked. Lots of people said it will never work, but it did. And in fact, I remember presenting this first result here in this same room 20 years ago. So in fact, we could reproduce the velocity of this Listeria with beads, and here we vary the diameter of the bead, and you see that we still have still obtain these trajectories by varying the diameter of the bead, but you also see that we have this what's called saltatory motion. So the bead moves by jumps, and in fact the interesting thing at that time was that these jumps had been seen with Listeria by applying a mutation on Listeria genes. And so we can just reproduce this, phenomenological difference just by changing the radius of the bead, so the physical characteristics of the system. So it's just an example where mutations can do the same thing as physical parameters. So this is my first trajectory and my first phase diagram in one dimension here. So we can measure the force as a function of velocity by sticking our bead on a needle. And this needle, because it's very thin, it's animated by Brownian motion, by thermal motion. So just by writing that the elastic energy of these fluctuations equal the thermal energy, we end up with this spring constant that will allow us to measure the force on this bead system. So the experiment was done by a very talented PhD student. So we have this needle that has been calibrated. So we know the spring constant, so we will be able to measure the force. Then to glue the bead on this needle, we put a drop of glue. We have the bead that is held on a micropipette here. So we put it together. Then we send a UV laser spot to cross-link the glue. And then we end up with this bead attached to the end of this needle. And then to cover this bead with the activator of actin polymerization, we just incubate. So this is in air. We incubate the solution of activator with this bead. And then we put it in a microfluidic system. And we end up with this experiment here, where you have the needle here, where the comet grows. It is held by the micropipette here. And so it was our first direct proof that that it's an elastic medium. So here you see the force as a function of time just before it breaks. So we pull on it, but if we pull too strong, then we break. The comet breaks, so it breaks at the surface of the bead, but it's still able to grow again because the activator remains on the surface. So just by analyzing this, this slope here of force as a function of elongation during these 10 seconds, we could derive the elastic modulus of this network, actin network. So it was one of the first direct measurement, direct proof that these networks are elastic. So we could go further in trying to understand the force-velocity relationship. So again, so we have an external force here that is imposed by our needle. Or I should say we have a feedback loop, so we can keep the force constant and measure the velocity that depends on this force. And so we stick our bead to our needle. This is the external force. We grow this actin network. The difficulty here is that this actin network grows itself. So it's not, it's an elastic that grows, that constantly grows at the surface. And so we have a velocity here. So we understand this as an elastic force that squeezes the bead forward, but also a friction force that is due to the friction between this network and the surface. And so we can through this tangential stress that we talked about in the symmetry breaking problem, we can derive the elastic force. So the elastic force reads like this, where Vp is the velocity of polymerization of the network, V is the velocity of the bead, R still the radius, and E the elastic modulus. And we also have this friction force. So here it's not a liquid friction force, it's a surface friction force, just because there are bonds that are attached and detached constantly at the surface of the bead. And these bonds generate friction, just like this. I have friction between my clay and the surface of the board. And so if we write this in this condition, so we obtain this line here. This is velocity as a function of force. And we can derive that the stall force, the force that is necessary to apply to stop this bead from moving, is of about 7 nanonewton. Right, and what we derive from this model here applied to these points is that the thickness is thickness that pushes on the side is on the order of 2 micrometer, which was the same before the movement. And the velocity of polymerization is about 1 micrometer, 1.6 micrometer per minute, which is the velocity, the classical velocity obtained in this system. So maybe some of you already thought that we may be in situations where we have limitation of diffusion, because this is a network. So here it's still our 2-color experiment. We start with green actin. We continue with red actin. And you see that what was spherical here before is sort of extended. So there is some reorganization in the network that is under stress all over the sphere. And so sometimes in certain conditions we may be limited by diffusion, because this is a network. The mesh size, I mean, depending on the mesh size, the monomers will have difficulties or not to diffuse through the network to reach the surface and be able to polymerize. So we have this phase diagram that depends on the elastic modulus and the diffusion coefficient. So if you have low diffusion, I mean a small diffusion coefficient, you will have a diffusion limited regime, because the monomers won't have time to polymerize at the surface. Whereas for higher diffusion coefficient, you will have regimes where what is limiting is the stress or the polymerization. So this is my second phase diagram. Okay, so now I will change. Where is Mathieu? Oh, yeah. Okay. So I'll go to some other experiments. So this time is so this is, in fact, what we obtain. So here we have a liposome, this time a membrane. So it's not a bead anymore. It's not a surface, a solid surface anymore. It's something that it's a membrane that can be deformable. And so we grow this network at this membrane. And what we observed was this. So at the beginning, we thought, oh, that's wonderful. Actin polymerization is able to deform the membrane. It's something active, etc. In fact, this was a serendipity experiment. So the master student forgot the sample under the microscope. So water evaporated. So in fact, our liposomes deflated. So it's not something active at all. It's purely passive. And this was, so we decided to redo the experiments in a controlled way. We grow the actin network around these liposomes. We stop the growth just by diluting the sample. And then once it's stopped, we deflate the liposome just by adding some solute outside the membrane. And in fact, this came with, after a discussion with Andrei Kosmanji at a Gordon conference, when he saw my buckling things and my wrinkling liposomes. So, well, I know what it is about. And it was solved in a quarter of an hour. And so in this control condition, the frequency of deformed liposome as a function of the thickness of the network. So maybe I should have said that in purple, you have the membrane. In green, you have the actin. So you see that if you increase this thickness, so you have more buckling when the thickness is small and more wrinkling when the thickness is large. So how do we understand this? In fact, it's a very simple mechanical problem. If we have thin actin shells, so the whole thing needs to deform during the deflation. And the only way energetically optimal to deform is to obtain this sort of mirror buckling. And where the energy cost is the highest is here. And it can be estimated here. Whereas when the thickness is larger, the whole thing cannot deform. It's too stiff to deform. And then what happens is that you have a reorganization next to the membrane that is very soft. And this, so this wavelength, so you have deformations here that create this ondulation. Nothing moves here. Everything reorganizes next to the membrane. And then you have wavelength that depends on membrane bending rigidity and the elastic modulus of the actin. And so we estimate this wavelength at 350 nanometer. And for intermediate thicknesses, well, of course, you have intermediate behaviors. So we put all our points in this diagram here. This is the thickness of the shell as a function of the radius of the liposome. And you see two groups, and we can extract the lambda, the wavelength, from these points, and we find 2 micrometer. That is slightly bigger than 350 nanometers. But so this was something that a reviewer helped us to understand. In fact, in our systems, if you remember from the symmetry breaking experiments, we are under stress. So because the network grows, it pushes on the liposome. So before we deflate it, the liposome is under stress. So this just increases this wavelength that we find at the end. So in other words, we deflate a system that is already that has grown under stress. So this was my phase diagram 3. Okay, so now let's come back to the dynamic system. So we use the same system with experimental system with a membrane here, but this time we don't stop the growth. So we look at a dynamic system. You have this branching mechanism at the surface, and we look at what happens. And the surprise was that we see tubes on a membrane that are being pulled during the mechanism of growth. So this is the membrane that you see. You see that this is where the network is, because this is the actin, and this is an overlay of both. So this is an image in phase contrast, and in phase contrast microscopy, you see that there is a thick thing, but you need this fluorescence experiment to see that we have tubes. So we checked that it was not an artifact of liposome formation. So if we destroy locally the actin, what we find is that, so of course the actin disappears, but you see that the membrane recovers its spherical shape. So it's really the actin dynamics that generate these tubes. So this result arrived in sort of dogmatic time in membrane deformation, because the textbook view of how membranes deform in a cell was, or still is for some of the people, still is, so that to deform the membrane, you need curvature proteins that assemble, generate a curvature, and only at that time actin starts to polymerize. And in fact, some other people were, so in fact, this was at EMBL in Heidelberg. So when I presented this result, people told me, wow, this is what I see in cells. So they had a careful correlation of fluorescence experiment to be able to see each protein one by one. And they were able to show that, in fact, before this before this cap forms, they could see that actin was polymerizing, that actin dynamics was able to initiate this bulge formation. And in fact, looking at the literature, there was also this experiment here. They take membranes from the inside of a cell. They put it inside the cell extracts that I defined earlier. And what they see is that if they don't put ATP, so I forgot to say that for all that to work, we need adenosine triphosphate to chemically change into adenosine diphosphate. And so we need the chemical reaction to obtain this. So if you don't put the chemical reactive, then you get a smooth liposome. But if you put ATP, then you get these tubes that we see. So this was with membranes from intracellular cell membranes, and this was with cell extract. And their conclusion was it's either lipid curvature or actin polymerization forces. And what we show here is that actin polymerization forces are able to pull these tubes. So because we know from physics that the force to pull a tube is punctual, so if you want to pull a tube of membrane, you just touch it and pull it with this force that is proportional to the square root of the bending energy and the square root of the membrane tension. So we thought if we decrease the membrane tension, we will increase the capacity of tubes to be pulled. But this is not what happened. Biology is often surprising. So if we decrease this membrane tension, we obtain other structures that were, I mean, again, that was a surprise. So we see that we generate what we call spikes that push the membrane inside the liposome. So I show you some images where you see, here you see these spikes, and here you see these tubes, another one here where you see these spikes. They are conical, they are often conical, and these tubes that pull away. So there are many ways of modeling this, and there's still a need to understand what happens. What we decided to do is treat the network as viscous. So it's what I call the viscous elastic trick. So we treat it as a liquid, and then we define the viscosity as the elastic modulus multiplied by the time, the viscoelastic time. I should say that all these networks are elastic at short time and fluid at long times. And there is a threshold of time, there is a critical time at which we can't consider them anymore as elastic, or we can't consider them anymore as fluid. So we use just this trick to be able to treat this problem. So there are other ways of doing it by kinetic models or by looking at this branching under stress, but what we developed with this one. And in fact, experimentally, you can see here that these, so here it's again the red and green. First we use red actin, then we use green actin. And what you see is that green actin grows at the inside of the cone, whereas the already polymerized actin is inside the cone here. So it's very different from other what's called philopodia formation, where you have parallel filaments that are able to push the membrane all at the same time. Here it's really a 3D cone that we build, and this is more, it's closer to what's seen in neurons, like dendritic philopodia. So in fact, we could, again, use our red and green experiment, and you see that in the, here it's green actin, you see that you have, sorry, it's red actin. Here you see that you have green filaments that emanate here. So we still have tubes that can grow either by green or by red actin. So constantly what we have, so again, to pull a tube, you need a point force. But this point force, this attachment can detach. And then other attachments to the network will take their turn. And so we have growth and shrinkage of these tubes within the network. So putting all that together, we could derive this phase diagram here. Well, we vary the mesh size by varying the protein composition. So you vary the mesh that characterizes the network. We vary membrane tension. And so we have either no tubes and no spikes, which was what we used to see before. We have tubes or no spikes, or we have tubes or spikes, depending on so the mesh size and the membrane tension. And this is my phase diagram 4. Right, so to wrap up this part here, I have shown you that we were able to reconstitute actin polymerization at the surface of an object by adding also capping proteins that prevent these filaments from continuing to grow. I have explained symmetry breaking, how we measure the force during this motility. I didn't have time to show you this. This is an oil droplet that is propelled by the same actin polymerization system. But here, instead of having a hard substrate, we have a fluid substrate. We have a fluid surface. And at this fluid surface, the activator, when it detaches from the actin network, can diffuse again to the front. So we have this saltatory motion because we have spontaneous diffusion of the activator to the front. So here it's moving, squeezed, then it will detach, and then the activator will move again to the front and squeeze again and detach, etc. And another thing I won't have time to talk about, so again, this is available, is this. When we add myosin motors here, so if we add myosin motors to this network that is growing, we can see the equivalent of what happens at the rear of a cell when under tension, but this time not by the geometry, but tension induced by myosin motors. We can see this breakage, cortical tension breakage. Right. So there are still unanswered questions, and I decided not to address them because the field was so dogmatic against the fact that actin could generate curvature of a membrane that I won't address them. So it's a sort of an open call. So we until here we considered the network as a mesoscopic system that grows at the surface at a velocity of polymerization, but the details of this growth have been deciphered by this very nice work by the group of Laurent Blanchoin. So what they do is they put an activated fiber, so only this fiber is able to generate the network formation. And you see that an actin filament here is growing. This red arrow is the end of the filament that's growing. But as soon as the filament that undergoes Brownian motion touches the activated surface, it creates a new branch. And this is what you see here. This yellow arrow is a new branch. And you see a new branch forms here, etc. So for this network to grow, you need a filament to touch the membrane, and then a new branch is formed. So it's a sort of branching mechanism at the surface. So we modeled it as a fluid surface that pushes the membrane, but it's much more complicated than this. And in fact, again, so this is a scheme that shows what happens. You need to have this activator. You need to have, I didn't go into the details, but this branching activator that interacts with this activator at the surface. And then you have a branch that forms that can be capped by capping proteins, so that at the end you have a network that grows by branching on an active zone, and you have a dead zone where everything is frozen because these branches cannot interact with the surface and cannot continue to polymerize. Right, so the question that remains is how to link this microscopic mechanism to the mesoscopic scale. In particular, so we know that the growth of this actin network can pull tubes, but in fact the way these conical shapes form is not clear. Well, so now I will end up with just a few slides about our new project here at LPENS. Just to remind you that we have looked at how this actin polymerization generates a force and a displacement of this membrane and the displacement of objects inside the cell and this retraction here. But in fact, I've never considered that the cell has a nucleus inside. So the new project here was an occasion to look at to give a new look at this cell motility by but this question, how does the cytoskeleton pull or push the nucleus during cell motility? So in fact, what is known is that inside our body, when cells move, they have to go through small holes of the extracellular matrix. And in fact, the nucleus is stiffer than the rest of the cell. So the nucleus is able to stop the movement of this cell because this is very stiff. So the cell needs to pull or push it for it to go through. And this happens all the time in our body because it's a three-dimensional network of collagen, for example, and the cells need to go through. So what we know from works that have been done is that the nucleus, as you know, contains the DNA. So underneath the nucleus, you have a shell here of lamin. It's a biopolymer. And this shell underneath the double membrane of the nucleus is linked to the actin that we talk about a lot. So we know that there are possibilities of actin to pull on the nucleus, and maybe with myosin to push on the nucleus. So in fact, what we decided to do is take microfluidic systems, so sort of, again, mimic what happens in physiological situations, mimic this in a microfluidic system made of pillars. And here you see underneath the cell that is going through. So you see it's squeezed through the pillars. And you see that the nesprine that we have marked, so we marked this protein here. So the nesprine goes to the front. So you have an accumulation of nesprine to the front, as if, but we are not sure yet, as if actin was able to pull the nucleus through. So here it's the same sort of cell goes through large avenues, and you see that there is no accumulation of this link to actin at the front. So this is a quantification here. So the result we got is that nesprine is enriched at the front. And this is an active dependent process, because if we aspirate the cell in micropipettes, we don't see the same accumulation at the front. So it's an active process that is due to the presence of actin, that is due to the fact that cells are going from the left to the right. So in fact, we decided to use this because this is an activity of cells, and when metastatic cells migrate very fast, they are more active, they are more active or aggressive. So how could we quantify this aggressivity or this activity? So we decided to use a Langevin equation that was used for another type of motile cells. So the principle, you know, is that the acceleration equals a deterministic term here and a stochastic term here that depends on the position and velocity. And here the stochastic term depends on position and velocity, and this is a random function. I mean, this is white noise. So our idea is to apply this not only to position and velocity, but also to polarization, to how this nesprine is accumulated at the front. So to run this Langevin equation on the variables that we have in our system. So this is an example during the lockdown, a try. This is only a try. That we have the velocity here as a function of x, and we derive from there the deterministic component. So you see already that here, this is the border of the constriction here that corresponds to the entry and the exit of the constriction. You see that we have an increase in amplitude of this deterministic term here before it enters and after it goes. But in fact, we're working on that for the moment. This is the face portrait of my talk. And so what we're doing for the moment is using this Langevin analysis for analyzing the trajectories. So this is the mass multiplied by the acceleration equals the driving force that we don't know, minus this friction force that we have talked about at the beginning of the talk, plus this random force here. So because we're at low Reynolds number, we think that we should use the overdamped Langevin equation that reads like this. So the x dot equals the driving plus this term here that takes into account the diffusion, the natural diffusion of the nucleus, and this white noise term. So I won't go into the detail now, but in fact, we decided to not use this acceleration term, but to use the overdamped Langevin equation instead of underdamped Langevin equation analysis. So now how do we define trajectories here? So this is what we did. So the barycenter of the nucleus is taken as the barycenter of the surface of the nucleus when it goes through constrictions. So we have an example of trajectory here. We decide that the start of the trajectory is when the center of the nucleus goes through the pillar preceding the constriction. And so you see that it goes here, it enters the constriction, it has some difficulty to enter a constriction, you have a plateau here, and when it goes through, it's very fast. So these are the trajectories that will feed the model, and this is a trajectory in a large avenue here. So I won't go more into the detail here. This is my trajectory slide. So I would, I hope I have created enough frustration that you will go to the PhD defense of Cyrine Amiri on the 12th of December. So Cyrine is my first PhD student here, and it has been a pleasure to start this work with her and with you here. So now I would like to thank the people who are the member of the teams on these projects. So Julie Plastino has been my collaborator for 20 years. And I'd like to thank her for her patience and her competence. And Cyril Amiri and Inge Boss have been accompanying us on this new project here. Our collaborators are great people, both from theory in physics, in microphysics, and in biology. And I'd like to thank all the people who helped us to shift to this new project. Patricia Davidson, who did the first experiments of cells in constrictions. Sofiane Mra, who during the lockdown tried to use this Langevin equation on our system, and Anna Nel-Mansouri, who helped us to start with different types of cell lines. So I thank you very much for your attention.